Hi, good evening, everybody. Welcome to Café Sci for August 2021. My name is Brad Peroni from Carnegie Science Center, and I am very excited for tonight's speaker. Now, before we introduce Dr. Balangota, I would like to remind everyone that we do have a question and answer box in the bottom of your Zoom window. So feel free to enter any questions you have during the talk into the Zoom Q&A box, and we will pose those questions at the end of the presentation. I would also like to thank our sponsors. Café Sci is presented by PPG and sponsored by Cook Myosite. But now it is my pleasure to introduce Dr. Anusha Balangota. Dr. Balangota is a postdoctoral researcher at the Department of Geology and Environmental Science and Pittsburgh Collaboratory for Water Research, Education, and Outreach at the University of Pittsburgh since 2019. She earned her PhD in environmental science from North Dakota State University. Her research examines nutrient dynamics, speciation, and sources, focusing on environmental controls of algal production and assessing causes and consequences of cultural eutrophication of freshwater systems ranging from lakes and reservoirs to urban streams and rivers. Her research employs a wide range of approaches from aquatic ecology, limnology, biogeochemistry, and hydrology. Dr. Balangota is working on the NSF-funded collaborative project assessing the ecosystem impact of drinking water orthophosphate addition on urban watersheds with researchers from the University of Pittsburgh and the collaborator Pittsburgh Water and Sewer Authority. With that, I will gladly turn things over to Anusha to start her presentation, and I look forward to seeing you all again for the Q&A. Anusha, it is all yours.
Thank you so much, Brad. So, I'm going to share my screen. Can you all see it?
Yep, we can see that just fine.
Yeah. Welcome, everyone. Thank you for joining me this evening. So today I'm going to talk to you about a collaborative project conducted at the University of Pittsburgh that assesses the effects of a large-scale drinking water orthophosphate addition on urban stream nutrient dynamics and limitations. So this project is a collaborative effort between Pitt researchers and Pittsburgh Water and Sewer Authority, funded by the National Science Foundation, with contributions from Pittsburgh Water Collaborative and Pitt Strides program. So I thank our collaborators at Pittsburgh Water and Sewer Authority for their assistance with drinking water samples. The drinking water contamination with lead gained much public attention after two high-profile incidents in US cities. For example, in Washington, D.C., the lead in drinking water crisis began between 2001 and 2004 due to a change in disinfectant from pre-chlorine to chloramine in 2000. Another recent incident was in Flint, Michigan from 2014 to 2016 due to switching water source from Detroit water to Flint River water, which did not have any added corrosion inhibitors. So in addition, other cities like New York City, Wisconsin, and Massachusetts also have lead contamination in drinking water. So let's see how lead crisis began here in Pittsburgh. Pittsburgh lead crisis gained attention in 2012. Then 2 years later, in 2014, PWSA switched, that means Pittsburgh Water and Sewer Authority, switched corrosion control treatment from soda ash to caustic soda without proper study or even notifying the Department of Environmental Protection. So in 2016, the PWSA switched back to that soda ash and found that city's lead level have increased above the EPA's action level of 15 parts per billion, first time on the record. The PWSA then started to add orthophosphate into Pittsburgh distribution system as a corrosion control chemical in April 2019 while replacing those lead lines. So since then, lead concentration in drinking water decreased to below the EPS action level of 50 parts per billion. So do you know that where this lead could come from? The lead could come from 4 primary sources when drinking water comes in contact with, such as lead soldiers, brass faucet and plumbing fixtures, lead goosenecks, and lead service line. So among these sources, lead service line contribute the most significant portion of the lead in drinking water, which is around 50 to 75%. So as we know that lead service lines contribute the most significant portion of the lead in drinking water. So when you look at this map, lead service lines are likely in use in all 50 states and Washington DC. Among these states, the deep red states, which is located in this Great Lakes area, have the most lead service lines. So there is no safe level of lead for both children and adults and affecting the whole body. Most notably, lead irreversibly affects intellectual development, behavioral processes, and overall life achievements of children. In order to control the corrosion in water service lines and meet the EPS action level, utilities use corrosion control in distribution systems. Phosphate-based corrosion controls are the most commonly used by distribution systems. When the corrosion controls are not in use, the lead leach from pipes into drinking water. However, when corrosion controls use, those chemicals reduce lead solubility by forming less soluble compounds, which form as a protective layer on the interior pipe wall and prevent pipe corrosion and lead leach from pipes to the drinking water. These orthophosphate-based corrosion controls have been used for around 80 years in US drinking water utilities, and most of the utilities now being shifted to orthophosphate. So in the figure here, I'm showing that when you have a corroded steel pipe, and then this is the lead pipe, and then when you have this phosphate treated lead pipe, so you can see this is the protective layer inside this pipe. So the PWSA, Pittsburgh Water and Sewer Authority, added orthophosphate into Pittsburgh water distribution system in April 2019. We know that our drinking water comes from the Allegheny River, and water is treated at the Aspinwall treatment plant to make sure that water is safe to drink. Then orthophosphate is added to the water distribution system at 3 different locations, which are distributed to different areas in the city to provide corrosion control protection before reaching customers. Orthophosphate is essential nutrient for the plants and microbes that make up the aquatic food web. However, since phosphorus is in short supply for most of the freshwaters, even in a small increase in phosphorus under right condition could make a whole change of undesirable events and accelerating the natural eutrophication. So algal blooms, especially if dominated by cyanobacteria blooms, can have serious environmental, social, and economic impacts, and including hypoxia, anoxia, and fish kills, and eventually shifting the algae composition and the nutrient limitation. So here the nutrient limitation means algae uptake nutrients in a stoichiometric ratio. However, algal growth can be limited by the available supply of either nitrogen or phosphorus, and eventually algal growth could drop due to the limited supply of that nutrient. In most of the freshwater ecosystem, phosphorus is limiting. So if you provide more phosphorus, you can see more growth. So in addition to orthophosphate used as a corrosion inhibitor, orthophosphate is an essential nutrient for the plants and microbes. Pittsburgh drinking water distribution system loses around 50 million gallons of water each day. This missing water could be due to unaccounted reasons, including leaks, small breaks, unmetered or occasional water main breaks, and likely to infiltrate into groundwater and could ultimately impact the nutrient status in urban streams. In addition, phosphorus concentration in domestic wastewater and greywater also could increase as water used for irrigation and city hydrant flushing. The target orthophosphate concentration at the customer's tap is 500 micrograms per liter. This is a relatively high concentration considering that algal growth could start as low as 10 micrograms per liter. That is why we are looking at, are there any impacts of phosphate treated drinking water on urban stream nutrient dynamics and nutrient limitation? This work is a highly interdisciplinary project that contributes researchers from multidisciplinary fields are working together with our collaborator, Pittsburgh Water and Sewer Authority. Despite the importance of using orthophosphate addition as a corrosion control approach to protect human health, there is a knowledge gap exists regarding the role of aging and leaking drinking water infrastructure on urban stream nutrient dynamics. So today I'll speak mainly about our results regarding the impacts of orthophosphate addition to urban stream nutrient dynamics and limitation on algal growth. So let's move into our research questions and hypotheses driving for this work. There are two research questions driving this effort. First, what is the role of leaking infrastructure on urban stream nutrient dynamics? And does orthophosphate addition shift nutrient limitation and eutrophication potential in urban streams? So we hypothesize that orthophosphate addition will change the chemistry and microbiome composition of urban streams. Especially we hypothesize that with orthophosphate addition, total phosphorus concentrations are expected to increase in urban streams due to drinking water losses from the drinking water pipe network and combined sewer overflows. In addition, with this extra phosphorus available in urban streams, we expected that could cause the streams to shift from phosphorus limitation to nitrogen limitation. So let's move into our sampling sites and methods. In Pittsburgh, most urban streams were buried in the stormwater pipe network. For this study, we collected samples from 5 above ground streams in Pittsburgh. So these streams, if you could look at the map here, and then this color code is represented with the location. Here the Shades Run, which is located in Allegheny River Boulevard. This is the Spinol treatment plant. And the Negley Run located in the Negley Run Boulevard. And Panther Hollow and Gibbs Run, they are located in the Shenley Park. And the Fern Hollow located in the Pitt Park. So we collected samples monthly from these 5 urban streams and 7 sites with the drinking water pipe network. That one is showing that DS1 to DS7. Those are the drinking water pipe sites. So the phosphate addition began in April 2019 and samples were collected from February to April 2019 to represent the precondition before the phosphate addition. Then samples were collected after the phosphate addition to represent the post condition from April 2019 to June 2020. So this monthly sampling was conducted during dry weather conditions to sample base flow rather than the precipitation influenced storm flows. So this is here I'm showing the surrounding land use types in 5 watersheds of sampled urban streams. Among these watersheds, Negley Run is intensively developed, and Phipps Run shows the most open spaces due to the golf course. And the Shades Run has evenly distributed land use. So when we compare with the Panther Hollow and Fern Hollow, they're moderately developed. So let's move into the methods used for this work. So to understand the water chemistry of these streams, we monitored on-site parameters and samples were analyzed for total and dissolved nitrogen and phosphorus, major ions, and trace metal in all 5 urban streams. In addition, PWSA is providing water chemistry data of 7 drinking water pipe sites. Additionally, we carried out a nitrate isotope analysis to identify the origin of source of nitrate in 5 urban streams. We also analyzed samples using qPCR and 16S rRNA community sequencing method to quantify if increased phosphorus exposure influences the microbial community in drinking water pipes and urban streams. However, today I'm not going over that part of the work, and that's going to be another great talk. So in my next slide, I'll show you more details of nutrient addition bioassay. So here we carried out nutrient addition bioassay. So we wanted to determine whether orthophosphate addition impact on nutrient limitation and algal growth. This is under laboratory condition. For the nutrient addition bioassay, urban nutrient samples were cultured with green algae. So these are, we purchased these samples. So this light green color, these are green algae. So the species we use is called Cyanodesmus. And this dark blue-green color, those are cyanobacteria samples. So there is a specific species we use, genus we use, this is Cylindrosperma. So we purchased these samples. And then after that, we collected samples before orthophosphate addition and after orthophosphate addition, those stream samples. So then those stream samples were inoculated with known concentration of pure cultures of this green algae and cyanobacteria. Then we spike with known concentrations of nitrogen only, phosphorus only, and nitrogen and phosphorus only. So we have these the Falcon tubes here, and then we use a series of nitrogen only, phosphorus only, and nitrogen and phosphorus only, as well as control. So we added the nitrogen, phosphorus, and nitrogen and phosphorus into these Falcon tubes with these cultured cyanobacteria and green algae. After that, those nutrient spike samples were inoculated for 7 days, and we measured the absorbance, which is a proxy for chlorophyll A concentration, and that is representing the algal biomass. So after we have these samples inoculated with these cultured cyanobacteria and green algae, and then we leave 7 days under grow lights. After the 7 days, and then we measured the chlorophyll A biomass. Now I am going to show you some results. Here, the y-axis of the figure is orthophosphate in concentration in ppm, milligrams per liter, and x-axis here is sampling month and year. So this is the box plot, shows the orthophosphate concentration in samples from 7 distribution pipe sites. So this is showing the orthophosphate addition in April 2019. So you can see here, there was no orthophosphate in our drinking water before orthophosphate addition. So let's see after orthophosphate addition, orthophosphate concentration in drinking water rapidly increased. However, it has been stabilized to around 1.5 ppm or 1.5 milligrams per liter. So this is the orthophosphate concentration in the drinking water before and after orthophosphate addition. So now I'm going to move into see phosphorus concentration in urban streams before orthophosphate addition. Here, y-axis shows the total phosphorus concentration. Total phosphorus means it includes both organic phosphorus and inorganic phosphorus. And then x-axis here is urban streams. So this box plot shows that total phosphorus concentration and trophic boundaries before orthophosphate addition in urban streams. So it is interesting that before orthophosphate addition, Negley Run was eutrophic and above the eutrophic threshold of 75 micrograms per liter. However, other urban streams, they were mesotrophic. So the phosphorus is the nutrient in short supply in most fresh waters. So even with the minor increase as low as around 10 micrograms per liter could negatively impact these streams. Here, y-axis is total phosphorus concentration in micrograms per liter, and x-axis I am showing the sampling month and year. So this is the time series plot showing that comparison of total phosphorus concentration in that means before the phosphate addition here, and then I'm comparing with after the phosphate addition here. So I'm comparing the same month, February, March, and April before orthophosphate addition with February, March, and April after orthophosphate addition in 5 urban streams. This shaded by showing the orthophosphate addition in April 2019. So you can see that total phosphorus concentration shows an increase then fall, the zigzag pattern throughout the study period for all 5 urban streams. So we hypothesized that orthophosphate addition would increase total phosphorus in these urban streams. So our statistical analysis between pre and post orthophosphate addition saw a significant increase in total phosphorus in urban streams followed by orthophosphate addition. In addition, we perform nitrate isotope analysis to identify the origin of nitrate in sampled urban streams, and also to determine whether this treated tap water actually reaches these urban streams. So here the y-axis is delta oxygen 18 and x-axis is delta N15. And this figure here shows the dual nitrate isotopic source plot for the 5 urban streams. So let me explain that these are the boxes. These boxes represent the ranges in isotopic signatures of nitrogen and oxygen for different nitrate sources. For example, here for the precipitation, here for the ammonia and fertilizer, here septic waste, soil likewise. So this is clearly showing that substantial wastewater nitrogen contribution in all urban streams. That means the wastewater is the dominant source of nitrate in these urban streams. So in here, we have this all 5 urban streams. You can see all of our samples are belongs to this box. That says that the wastewater is the dominant source of nitrate in these 5 urban streams. Here, I'm showing the y-axis. Here is Tn to Tp ratio. So we use this Tn to Tp ratio. That means total nitrogen to total phosphorus ratio, which used to interpret nutrient limitation status. So here we have a box plot. It shows the total nitrogen to total phosphorus ratio in all urban streams before and after orthophosphate addition under the natural environmental conditions. So based on our TNTP ratios, our urban streams were phosphorus limited before the phosphate addition. And then we hypothesized that urban streams would switch from phosphorus limitation to nitrogen limitation followed by orthophosphate addition. However, we found that there is no significant change in nutrient limitation in urban streams following this orthophosphate addition under natural environmental condition. So in my next slide, I'm going to show you the direct influence of nutrient addition on urban stream nutrient limitation using algal bioassay that is carried out under laboratory condition using the water from these 5 urban streams and tap water. So I'm going to here show you those results from algal bioassay. Here we use 2 groups of phytoplankton, we call cyanobacteria and green algae. If you still remember that I explained the how we did the nutrient addition bioassay. I explained that we use cultured green algae and cyanobacteria. So this is the results from that. So the y-axis here is absorbance, which is a proxy for chlorophyll A, and it represents algal biomass. And then x-axis here, I have my treatments. So I have control here. That means I did not add any nutrients. And then here I have the 4 treatments. I added phosphorus, and then I have 4 treatments here for nitrogen. Those are added nitrogen, and I have 4 treatments here for both nitrogen and phosphorus. So the figure here shows the cyanobacteria and green algae biomass in response to those treatment before the phosphate addition is showing the blue color, and the 2 months after the phosphate addition showing the red color, and 12 months after the phosphate addition showing the green color for tap water. For this nutrient addition bioassay revealed that in tap water, nitrogen alone addition, that means only nitrogen addition, caused a significant increase of green algae and cyanobacteria biomass than phosphorus alone addition. That means only phosphorus addition, and both nitrogen and phosphorus additions following 12 months after phosphate addition. So this indicates that nutrient limitation status changed from nitrogen and phosphorus co-limitation to nitrogen limitation 12 months following orthophosphate addition in tap water. So in my next slide, I'm going to show you what happened to the nutrient limitation status in urban streams. So in addition to tap water, some urban streams, including Shades Run, Fern Hollow, and Panther Hollow, also showed the N limitation, that means nitrogen limitation, however, only 2 months after phosphate addition. And then I'm not showing the data here. And other urban streams like Pittsburgh Run and Negley Run, those streams were either NP co-limitation or did not respond to our nutrient additions. So let's move into our conclusion and discussion. So we have observed a significant increase in total phosphorus concentration in urban streams following orthophosphate addition. In addition, we also observed wastewater is the dominant source of nitrate in the 5 urban streams sampled in this study. So this finding further suggests that orthophosphate in these streams may also be derived from wastewater. So under natural environmental conditions, we did not observe a significant change in nutrient limitation following orthophosphate addition with a test that we compared all urban stream values before and after orthophosphate addition. However, we have nutrient addition bioassay, which was under laboratory conditions, indicates that a significant change in nutrient limitation following orthophosphate addition in some urban streams and tap water. So when we are discussing these results, if you notice that among these urban streams, Negley Run behaved differently. So let's see why. So Negley Run was even eutrophic even before the phosphate addition. And then we have our land use map. So based on this, Negley Run watershed is intensively developed than other 4 watersheds. In addition, we have carried out this nutrient addition bioassay. So if you could still see the colors, so this is my control. That means I did not add any nutrients, and this is the one that I add nutrients. You can see the same green color. What does mean that? That means the control means without any nutrients. So the Negley Run is already rich with the nutrients that required for algae to grow than other urban streams. So that is giving me sense that Negley Run is already highly eutrophic, and then if we add more nutrients, it can be more eutrophic in the future. So there are important implications of this study. So this is the first study to investigate the impacts of large-scale drinking water orthophosphate addition to urban streams. In addition, this study covers the first step towards understanding the subsurface connection between the urban streams and urban infrastructures. Additionally, we also understand that orthophosphate is needed to mitigate lead. However, more pilot studies are needed to understand the lowest possible orthophosphate concentration that requires adding into distribution system to optimize protection of human health while minimizing environmental impacts. That means we want to do more studies to understand what could be the potential orthophosphate concentration that we can add it into distribution system while minimizing the environmental impacts because of these failing urban infrastructures and other unaccounted reasons of treated drinking water losses. So thank you everyone. So now I'm happy to answer any question you may have. In addition, I would like to advertise that we have this type of talks we are doing at Pittsburgh Laboratory for Water Research, Education and Outreach. And we have the website here if you'd like to hear those kind of STEM related talk. And then I would like you to sign up for future talks. Thank you.
All right, thank you, Anusha. So we already have a couple of questions coming in, but I'd like to remind you all, you can put your questions in the Q&A box found in the bottom of your Zoom window. So while you are thinking about your questions, we will get started with the ones that are already here. The first question is, and Anusha, you are welcome to leave your shared screen up there so that people can see the address for the collaboratory if you like, or you can stop sharing your screen, your choice. The first question is it correct to assume that replacing all the lead pipes is prohibitively expensive, hence the orthophosphate addition?
Yes, replacing all the lead pipe is around 45 billion. It is very expensive and it is not economically feasible. So that's if one can replace all the lead lines, that is the ideal situation. However, that is not economically feasible. So until we get there, we want to control this the corrosion. That's why we start to adding orthophosphate to mitigate that, but that is not the perfect situation. Perfect situation is replacing all the lead lines, but understandable, it's very expensive.
Mm-hmm. And I'll exercise my host privileges here to follow up with a question of my own. What sort of time frame does this orthophosphate addition work on? Is this a permanent addition to the water supply that needs to continuously work on the lead pipes, or is this a time-limited solution?
What the PWSA is doing, actually, they started with a relatively high concentration at the beginning. However, they have to adjust their orthophosphate concentration because it's depend on the water chemistry, because it is highly pH dependent. So based on the pH level of the water, and then they have to adjust the orthophosphate concentration. That's why I use, so that there's a kind of rapid increase, and then once they understand that how much of orthophosphate they need in this distribution system, the concentration dropped into around 1.5 ppm, then that level has stabilized. That is in the distribution system, but in your kitchen tap, it's going to be low.
Okay. Does orthophosphate have any pathophysiological effect on humans?
I do not know. What I know is orthophosphate is a food additive, and then based on the FDA, it is generally non-toxic, because if you drink a can of soda, then you get more orthophosphate. But I do not know any human health-related impacts.
Okay, good to know. Good question. Why did you choose the particular streams that you chose for your study?
Yeah, because the Pittsburgh, most urban streams were buried inside the stormwater pipe. So we wanted to use the above ground streams. So those are the streams that were located within these distribution sites pipes, we need to have the nearby and then above ground streams. So that is the reason we selected those 5.
Okay. And another point here about the effect of orthophosphates on humans, we have Sarah Haig here says no impact on humans as the concentration is extremely low compared to other sources. So just following up on your previous answer. One more question we have here. You said that your study is the first to investigate the effects of orthophosphate addition. So no one thought to test the environmental impact before adding it. Is that to say that you're the first one in Pittsburgh or the first overall to study the impact?
This is the first overall because our study is simultaneously adding the impacts into urban stream network and the microbial communities. So this is the first study simultaneously added. There was one study but wasn't simultaneously studying the impacts on the urban stream network and then microbial community. So this is overall the first study. Yes.
Okay. So your study happens after it's first added to the water supply. So were there any, there were no laboratory studies that might simulate what might happen out in the wild before it was added either? Is that true?
And no, there were a pilot study did at PWSA because they were doing the pilot study to make sure that what range of orthophosphate they need to add into their distribution system. So they haven't done into the urban stream network, but they have done the pilot study to figure out that how effective this orthophosphate to control the lead contamination in drinking water. So they have done that before they add it into our drinking water distribution system.
Okay. So we have a question here. I did not understand the nitrogen phosphorus relationship completely. Would reducing nitrogen lessen the need for orthophosphate? So maybe you can clarify that relationship for us.
The relationship means the nitrogen to TP.
So Bill, feel free to follow up with
yeah.
a bit more detail if you want. I'm wondering if he's not referring to the slides that showed the spike in growth with the nitrogen and nitrogen plus phosphorus versus the just phosphorus additions. But yeah, so Bill, if you want to clarify that for us. So Bill is talking about the eutrophication effect.
The eutrophication. Okay, yeah, the eutrophication. So let's see that if you see the algal growth, so the nitrogen and phosphorus both need for their growth. So the eutrophication is a process. It's a natural process. But what we are doing, we are adding more nitrogen and phosphorus through anthropogenic activities to accelerate that process. So the when the water body is leading towards to eutrophication, there are other consequences like decrease in distal oxygen concentration and fish kills, and then having the most scums on the surface water and then reducing the light penetration. Those are the consequences of eutrophication. But in order to eutrophication is generally because of the enrichment of both nitrogen and phosphorus, because the algae and other macrophytes, they need both nitrogen and phosphorus because those are the two primary main nutrients that require for them to grow. So if you have one nutrient is limited, that means short supply, and they cannot then what happened, there are different type of phytoplankton, they may get grow, but initially how the eutrophication started with more nutrients. You need the more nitrogen and phosphorus. That is the nutrient enrichment causing the eutrophication. So that is the process. It need to build up those nutrients in this water body.
Okay. And so, and all of that is about the eutrophication in the streams, but that does not really impact the effect of the orthophosphates on the drinking water pipes, on the lead pipes. So the nitrogen isn't something that we need to think about as far as the lead pipes, correct?
So when you have the drinking water pipes, you have those phosphate. Those are phosphate treated drinking water. But if you still remember that, so the here Pittsburgh, we have most combined sewer overflows. And then I showed that my nitrate isotope source plot, and then most of the nitrate in our urban streams coming from those sewage because of those combined sewer overflows. So we have that nitrogen coming from, and then our drinking water pipes, we have those around 50 million gallons of water per day due to unaccounted reasons. So when you have this phosphorus, and then you have nitrogen also coming, so you are making that the adequate amount of nitrogen and phosphorus for them to grow. So if we have more nutrients coming with different the unaccounted reasons, and then you will ended up with having eutrophic conditions. So for best example is Negley Run. It's already have more nutrients.
Perfect. Perfect. Thank you for clarifying that for me. Sarah also adds another comment here that lab scale work was done to assess impact in several studies across the country, but this was the first full scale system study. So thank you, Sarah, for that. The next question is about what happens when this water reaches the end of the pipes. When the water gets to Alcosan, is there any technology to remove the phosphates from the water before that water is returned to the river?
Yes, because when water reaches to the Alcosan, so they have to remove the phosphate before dumping to the rivers. So that is something Alcosan has to do, because those wastewater treatment plants, they can remove most of the phosphate out of this wastewater. But here, what I'm talking about, so there are the drinking water pipes network. Because of this failing infrastructures, there are unaccounted reasons like small breaks, leaks, and water main breaks. So because of those reasons, those phosphate treated water can infiltrate into groundwater. So that is not the only reason. So we're not using those phosphate treated water for those the household activities. We are using for the city hydrant flushing and lawn irrigation. So then eventually we are going to increase phosphate concentration in wastewater and grey water. And with this, and the other reason is we have combined sewer overflows. So with this combined sewer overflows, not all of these water is reaching to Alcosan. Before reaching to Alcosan and treat there, we have those overflows and already infiltrated into groundwater and nearby urban streams.
All right. So this question says, on one of your slides, you showed data for nitrogen sources using isotopic analysis. Can you explain the NO3 from precipitation? Do you mean precipitation from atmospheric precipitation in rain or snow, or precipitation from insoluble salts?
Atmospheric precipitation.
Okay, perfect. Mark that one as answered. Can we study the change in medical problems before and after the orthophosphate in drinking water in a selected region? Are there scientific reports about the effect of inorganic phosphate on cardiovascular diseases in humans?
My answer is I do not know about the cardiovascular diseases related with the orthophosphate.
Okay. And if anyone out there knows of, medical professional who might have some research or connections about the effects of orthophosphates and cardiovascular diseases and inorganic phosphates, feel free to send them my way. I would love to invite them to do a future Café Sci to follow up with the environmental impacts of orthophosphates in our drinking water. So we can look at this from multiple angles, but we don't seem to have that answer this evening, and that's okay. We can't expect to have all of the answers all of the time. So we have plenty more questions we can ask, though. And the next question is, I think I saw Sarah chime in with a comment here. Renal specialists and cardiovascular specialists that Sarah spoke with at UPMC said the concentration in water will likely have zero impact as the concentration is very low. So that sounds like a perfectly great answer for this specific instance, but maybe still I invite an expert in the medical field to answer what if the concentrations someone exposed to are a lot higher for that maybe come from other sources, but it looks like the concentrations we have here are generally considered safe.
Yeah, but I can add something that I saw in the FDA saying that phosphate is a food additive. So if you drink a can of soda, the amount that you are going to consume is equal to the a few gallons of drinking water. So that means they're saying that there is no risks to having the orthophosphate because phosphate is used for most of the food grade preservatives, but I don't know especially for the cardiovascular.
Okay. So the next question, are the orthophosphates used in our drinking water organic or inorganic? Are they easy for plants to absorb in the form used by PWSA, or does it adhere to stream surfaces / sediment in the way that it does the pipes? And is there something in the natural stream systems that could change the behavior of those orthophosphates?
Yeah, orthophosphate is an inorganic, dissolved, readily bioavailable, and highly reactive. So that is you can find in stream water. So the orthophosphate that used in the distribution system is dissolved inorganic. It's readily bioavailable.
Thank you. And we have one more question submitted, but we do still have time to take any other questions. So if you'd like to put in a follow-up question or a new question, we do still have time to take a few more. So don't hesitate, don't feel shy. So the next question is, what happens to the phosphate that is removed? And I'm assuming that means, at the end of the line through Alcosan, or maybe you mean that's the phosphate that gets removed by algae, absorbed by algae, and gets fed into the ecosystem. But what happens to that phosphate?
I'm just trying to think about the remote. Alcosan is. I mean they have to remove phosphate from the wastewater because when they are treating, so there is a way that they can effectively remove much of the phosphate through their treatment process before they dump into rivers, because they have a permit that how much of phosphate they can dump into the waterways. So they cannot dump all the phosphate. So they are removing that. but in the, I'm not much clear that we are talking about before those wastewater reach into Alcazar. So we're talking about the phosphate leak from those drinking water pipe, and then phosphate can leak from those combined sewer overflows. Those are untreated. Those have phosphate. So when those are reach into urban streams, the LJ is going to absorb, uptake. That's correct. But if they are reached to Alcazar, then most of them are going to remove to this treatment process.
Great.
I
And
would
we
like
have a
to ask, yeah.
we have a clarification here to the question. So the question was, what happens to the phosphate after it's removed from the water at Alcosan's end? So does that end up in a landfill? Does that find some other ultimate destination?
I do not know what they are doing over there after they remove whether they are going to make it fertilizer. I do not know exactly what they are doing over there.
All right. If there's anyone from Alcosan on the in the audience this evening, feel free to drop a comment in the Q&A box, and we can get this person in touch with you to answer that question in a little bit more detail. A few more questions coming in. So it's been written that one possible contributor to the fall of the Roman Empire was lead poisoning from their plumbing. The Romans even added lead acetate, also known as sugar of lead, to food as a sweetener. The chemical abbreviation of lead, Pb, is from the Latin plumbum and is the source of our word plumbing. Just a comment. So yeah, interesting to think that we've been dealing with lead in our water for millennia, and somehow we're still dealing with it. So hopefully we will be able to turn the corner and get some more positive movement on the lead problems in our water.
Nice comment.
Another question here. Does orthophosphate change the lifestyle and weight of fish? A recent report has demonstrated that the level of inorganic phosphate is increased in cancer tissue in animal models.
I can't answer. I do not know orthophosphate caused because I haven't done that work, how they are bioaccumulating in the fish, but I know that heavy metals are bioaccumulating, but I do not know orthophosphate how these are going to make the cancers for the fish.
Okay, so if anyone out there knows an expert who might want to come and do a Café Sci talking about orthophosphate's effect on fish, please feel free to email us at the Science Center and let us know. But maybe you can say, clarify just a little bit about the indirect effects of orthophosphate on the effects of eutrophication in streams on fish populations.
Yeah, that's I can say that. I mean, that is obviously that I showed you the one of the slide that in the eutrophication, when you have the orthophosphate and nitrate, those are nitrogen and phosphorus. Those are going to increase the nutrient enrichment and then ultimately affecting the algal growth. So when you have the more algal growth, and what happened that they are making those surface scums and reducing the light penetration. And then what happened in the near bottom layers, the dissolved oxygen concentration is going to decrease. And then that can make the hypoxic and anoxic condition and lead it to the fish and other microinvertebrates who live nearby the bottom area and the water column. So they're going to die. Then we can see some fish kills, especially in the summer, due to this anoxia and hypoxic condition, because they require at least 2 milligrams per liter dissolved oxygen, and that has to be minimum at least 5 milligrams per liter for survive the fish, and that depend on the fish species too.
All right, thank you. And we have one, it appears to be final question, unless someone else wants to sneak another one in here before we finish. Is there any research on how phosphate affects the biology of the larger river systems, Allegheny, Monongahela, Ohio rivers? Is the phosphate concentration from drinking water negligible when compared to the runoff that flows into the rivers during storms?
We have actually we were doing study with the Ohio River. We were analyzing some data, but we unfortunately do not have the orthophosphate data, but we do have the total phosphorus concentration. So the negligible means that when phosphorus coming through the agricultural watershed, so then we can expect that more phosphorus can lead into those rivers through the agricultural watersheds than these urban streams. But we do not have the orthophosphate data, but we do have total phosphorus. But we are seeing that the total phosphorus concentration using those historical data set is gradually decreasing. So there is no increase in trend of total phosphorus especially in the Ohio River.
All right. So I see no more questions here. So I would like to take a moment to thank you, Anusha, for being with us this evening and presenting your work to our audience. I found it personally very fascinating and riveting, and this is something I'm sure that not everyone even knows is happening with our drinking water as a way to mitigate the lead problem that we have here in Pittsburgh. So thank you for shining a light on that. I would also like to share that several people entered their thanks into the Q&A box as well. And Sarah chimed in with one more comment that phosphorus is normally removed in the sludge in wastewater treatment plants and can be mined as a nutrient resource, so may be used commercially as a fertilizer product. So sharing those thanks from our Q&A box, I extend my thanks to you on behalf of the Science Center, and I would like to invite everyone to Café Sci next month, that is September 6th, to hear from Carla Rosenfeld. Carla is the Assistant Curator of Earth Sciences in the section of Minerals and Earth Science at Carnegie Museum of Natural History. Thanks again, Anusha. Have a wonderful night, everyone, and we'll catch you in a month. Stay safe, everyone. Be well.
Thank you, Brad.